It's a real pleasure to be here. It's very exciting to visit Indonesia for the first time. I'm a little bit sad that I didn't make it here quite in time to watch the Australia versus Indonesia football match last night, which was apparently very exciting game and a good result for both teams, I guess. So, yeah, I want to start by introducing my wonderful team, the Genomic Technologies Lab from the Garvan Institute in Sydney, including Tia, who's actually a PhD student in my team. So it's a really great team that brings together a diverse set of expertise in molecular biology, genomics, computer science, and sort of data analysis. We work on a really broad range of topics, but long-read sequencing technologies are pretty central to everything that we do. And in particular, nanopore sequencing, which is obviously the topic of today's meeting, is something we've been working on for a long time now, and sort of methods development in the nanopore sequencing space, software engineering in the nanopore sequencing space, and applications of nanopore technology for precision medicine is really a key focus of the group. Now, it's not just because nanopore is a cool technology that's kind of fun to play with. That's not just the reason we work with it. Really, the reason we are so interested in nanopore sequencing is because we believe it's a technology that has the potential to really transform and accelerate the way in which we diagnose inherited disease. And I guess this goal, building the tools and capabilities to make that a reality, is a key motivation for everything we work on. Now, given the topic of today's meeting, I don't need to introduce the mechanism of nanopore sequencing in great detail, I don't think, but I think suffice to say, it works by transiting a DNA strand through this protein pore that's embedded in a charged membrane, and that generates the electrical current data that we can translate into DNA sequencing information in real time. But the talk that I'm going to present today focuses on a particular unique capability. It's quite nifty capability of nanopore sequencing, which is called read-until or adaptive sampling. And this is basically a trick that allows you to program a nanopore device to recognize a given DNA strand as it is entering and moving through that pore. And then based on its sequence, you can sort of dynamically reverse the charge on that pore to spit out the DNA strand if it's not something that you're interested in. And what that means is that, in theory, we can now perform targeted sequencing in a fully programmable manner. So all of the target selection of which genes you're interested in happening right there on the device without any additional molecular methodologies to select for your target sequences that you're interested in. So when this was first sort of announced by Nanopore, we thought it seemed really like a pretty cool application. And it's easy to imagine how you could use this for many different areas, particularly in clinical diagnostics. But we wanted to know whether this sort of really works, because sometimes these things are a little exaggerated, and so we sort of set out to test this capability, and we wanted a specific application to sort of test this on and see whether it worked. And the first example that we chose centers around these specific types of sequences called repeats and repeat expansion disorders. So by way of background, the human genome contains lots of these so-called short tandem repeats, or STRs, which I'll use throughout the talk. And this is essentially just a short DNA sequence motif that's repeated consecutively again and again. So CAT, 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 all in succession. And these are quite common. They make up around 7% of the human genome, and they're highly polymorphic, which means they vary in size between different individuals. So we might have one individual who has, say, 8 copies of this motif, another individual with 11 copies, and so on. But they're of particular interest because beyond that normal genetic variation between individuals, these repeats can become greatly expanded in particular individuals. And these expanded repeats actually are known to cause disease. They're the known pathogenic variant in many different diseases. It's quite a complicated story. These repeats are associated with many different diseases. For whatever reason, a lot of these are neurological disorders, and this includes some quite well-known examples such as Huntington's disease, Fragile X syndrome, motor neuron disease, and many more. In fact, by our count, there are at least 37 there are at least 40 known neurogenetic disorders that are caused by repeat expansions in at least 37 different genes, which are shown in this table. And actually, this is now quite out of date since 2021. There's been many new repeat expansions described. And so this list is sort of growing all the time. Now, because these are long, highly repetitive, often very GC-rich sequences, STRs are very difficult, sometimes impossible, to analyze using short-read next-generation sequencing, so from Illumina or BGI, the sort of more established technologies. And this means that the diagnosis of these repeat expansion disorders still relies on some pretty old-fashioned hands-on molecular biology techniques, such as repeat PCRs, Southern blots, and some other techniques. Now, these are really good techniques. They work well, but they're quite hands-on, they're quite slow, they're quite labor-intensive, and what's more, each test only tests for a single gene, so it's like a one gene, one test kind of paradigm. And that's a bit of a problem because we have, at least 40 different genes here that are potentially in play. So a one gene, one test paradigm is not going to work very effectively for this particular space. So we thought this was a really good case for us to use our nanopore sequencing and deploy this read until or adaptive sampling functionality to programmatically target every single one of these repeat expansion genes in a single test, a single sequencing assay. So we basically designed a read until or adaptive sampling panel that has all of our STR genes, and we use this to perform targeted sequencing on the handheld MinION sequencing device. And here I'm showing you basically the sequencing coverage across a single gene, HTT, which is the gene that causes Huntington's disease. And by comparison to the sort of off-target regions, which are shown in red, you can see we get this nice enrichment of sequencing coverage across the body of the gene. So this is working really well. We're sequencing more of the gene and less of the rest of the genome that we're not interested in. And I just sort of mentioned that this work was done a couple of years ago, and we were using the sort of handheld MinION device. Now we're using the slightly larger P2 Solo device, which you guys just saw in the last talk. And this makes it possible for us to sequence, say, up to 4 patients on a single flow cell using this adaptive sequencing approach. So this looks really good. But what we're actually interested in is obviously our repeat sequence. And so at a specific position within that gene, HTT, there is a CAG repeat that has around 20 copies in healthy individuals. And so if we get sort of 30 to 40-fold sequencing coverage depth on that gene, we might get about 30 reads that fully span that repeat. And we can sort of phase these reads out into the maternal and paternal alleles, and then we can assemble a consensus repeat sequence for each allele. And so you can see for this patient, we've assembled one allele that has 64 copies of this CAG repeat, which is well into what is considered the pathogenic size range for this gene. So unfortunately, this would be a positive diagnosis for Huntington's disease. So that was kind of one patient. But in the next slide, we've genotyped the HTT repeat sequence across 12 different individuals, and 5 of which are known to have Huntington's disease. So you can see that this repeat sequence that we've assembled is very diverse. We get lots of different alleles. Every individual essentially has a unique pair of sequence alleles here, and they're covering a range of different sizes. But you can see that the 5 individuals who have Huntington's disease have a very clearly expanded repeats that are all longer than that known pathogenic cutoff for this disease, whereas our 7 individuals who don't have Huntington's all have shorter repeats that are shorter than that pathogenic cutoff. And so in this example, our test is very clearly and correctly distinguishing the normal from affected individuals, which is exactly what the test is designed to do. And when we sort of compare our assay to the standard molecular testing approaches that I mentioned, these sort of old-fashioned one gene, one test methods, across many different genes and many different patients, the results are highly concordant, which is to say that when we measure the size of the repeat with our nanopore assay, we're getting a very similar size to what you would get with the established diagnostic technologies. Now, for Huntington's disease, this is good, but it's maybe not necessarily a game changer. But for some of these other disorders that have more complex repeats than that simple CAG example, we really start to see the advantages of this long-read sequencing approach. So I'll quickly touch on two of these disorders next, Fragile X syndrome and Canvas, to give you a taste of what I mean. So in the case of Fragile X, the nanopore data allows us not just to measure the repeat and the size of that repeat, but also to determine the DNA methylation profile of the FMR1 gene, which is where this repeat occurs. And better yet, we can do this in an allele-specific fashion. So in this subject here, we see that the expanded STR allele, unlike the normal STR allele, is hypermethylated and silenced, and this epigenetic silencing mechanism is actually central to the pathogenicity of this repeat expansion in Fragile X syndrome. And so being able to identify this silencing mechanism in addition to the repeat size and in an allele-specific fashion in a single assay without using multiple different techniques is really a key advantage of this technology. The next disorder that I'm going to talk about is an ataxia known as Canvas. So this is a movement disorder that's characterized by loss of muscle control that can have quite devastating consequences for the patient. And this is caused by an STR expansion in a gene, RFC1. So RFC1 expansions are relatively common among the normal population, and they can be very, very large, sometimes around 5 kilobases in length. And they come in basically a bunch of different flavors. So it's a 5 base pair repeat, a pentanucleotide. In healthy people, you generally see either one of these 2 repeat motifs that are shown here in pink or orange, which are considered to be non-pathogenic regardless of how large they are. So they don't cause disease if they're pink and orange motif. Then the main pathogenic repeat motif is this 3rd, more rare sequence motif shown in blue. And if you get 2 copies of this pathogenic sequence, that results in Canvas, so it causes disease. And so for the genetic diagnosis of Canvas, we need to know several different pieces of information. We need to know, is there an expansion present in RFC1? How large is the expansion? We need to know whether the expansion is on one allele or on both alleles. And we need to know what is the repeat motif. And again, we need to know that on both alleles. And so to answer all of those questions reliably, you really need multiple different tests, and sometimes it's not even possible because this is a very challenging and large sequence. And so the question was whether we could do this with our read until assay, and I'll start by showing 2 subjects. So these plots here show the density of each nucleotide as well as each of our different motifs that was observed across the length of this STR site. So in the subject up on the top, who's healthy, we've detected 2 copies of that pink motif, both of which are around 600 base pairs in length. In the subject underneath, who has Canvas, we've detected 2 copies of the blue motif, which is that pathogenic motif, one of which is around 2.5 kb, another which is around 3.5 kb. And so this would basically be a positive diagnosis for Canvas. And you can see it's quite concordant with the southern blot test over to the right, which is the sort of established molecular method, where we see kind of 2 sequencing bands corresponding to those 2 different expanded alleles. But now I bring in like a 3rd subject who also has Canvas. And this person has 2 large expansions, both of which are around 5 kilobases in size, and they show up as a single band on this person's southern blot over to the right. This person had had a separate PCR test which confirmed the presence of the blue motif. And so this basically looks like they have 2 copies of that expanded blue pathogenic sequence, which would be a positive diagnosis for Canvas. However, when we sequenced this using our nanopore sequencing and we phased out the reads, we actually found that one allele over to the right is not the pathogenic sequence, but corresponds to the orange motif, which is currently considered non-pathogenic, so non-disease causing. But this is really quite interesting because this person has Canvas. So by the conventional wisdom, they shouldn't have Canvas because they don't have 2 copies of the blue sequence. And so what that tells us is that most likely this orange motif, if it's expanded beyond a certain size, can in fact be pathogenic. It can cause disease. And I guess that our assumptions that it's always non-pathogenic may have been misguided by some ambiguity in this molecular testing, which as I showed here, the old school molecular testing couldn't reveal the full picture in this case. And so I think that's a really nice, clear example of how long-read sequencing can enhance the resolution and improve the genetic diagnosis of these really complex repeat disorders. So we published that work as kind of a proof of concept 2 years ago, and since then we've been actually trying to use this method to improve the diagnosis of patients with neurogenetic disease. And essentially we're trying to demonstrate how this can be applied clinically. So one area we've been working on is for the diagnosis of spastic ataxia spectrum disorders. Canvas is one of these, but this is a group of inherited neurological diseases which manifest in progressive loss of muscle control, and this is really devastating for patients, as I said. So spastic ataxias affect around 1 in every 10,000 people, and they're quite heterogeneous. And in terms of the symptoms and the age of onset of those symptoms. And we know that there are over 450 different genes involved in this spectrum of disorders, as well as all different classes of genetic variation, ranging from single nucleotide variants, small indels, larger structural rearrangements or SVs, copy number variation or CNVs, and of course STR expansions, many of which are implicated in spastic ataxia. So as a result of this, in Australia, patients who are affected by spastic ataxia disorders, they often have to undergo many different sequential genetic tests. So they have different tests that assess different genes or assess different kinds of variation. So for example, a patient might have whole exome sequencing to identify sequence variants, followed by repeat primed PCR on a specific gene to look for a possible STR expansion. Then they might have another technique known as MLPA to look for structural variation, large deletions or copy number changes. And this process is quite slow and costly. More importantly, this kind of fragmented testing landscape means that many diagnoses are potentially missed because the right test wasn't necessarily ordered for the right patient at the right time, or perhaps the right test wasn't even available to a particular patient. And so we set out to try and, I guess, change this by designing a targeted nanopore sequencing assay that would capture all 469 genes that are currently known to be implicated in spastic ataxia, and leverage the capabilities of the long-read sequencing to simultaneously detect every class of genetic variation across every one of these genes, all in a comprehensive integrated assay. Now, to evaluate this kind of single test approach, we've so far applied this new method to a cohort of 34 genetically undiagnosed patients. So these are patients who have spastic ataxia in a phenotypic sense, but have no previous genetic diagnosis identified. And to summarize very briefly, this has worked really quite well so far. Our single test approach has so far produced a new genetic diagnosis in 41% of these patients. So this is in patients who've had no previous genetic testing. But more strikingly, in those patients who had already been through standard genetic testing pathways in Australia, but remained undiagnosed, we've found a new diagnosis in 39% of these patients. So this is a really impressive uplift in the diagnostic yield, and that's a very encouraging result. There were kind of several explanations for these new diagnoses. So in some patients, we detected an STR expansion for which there was no current test available in Australia, specifically for these genes FGF14 and RFC1, one of which I mentioned a second ago. Or perhaps where a test was available, but it was not ordered because it was not considered to be a likely cause for that specific patient. Outside of STR expansions, we found small sequence variants in genes who had not been considered during previous testing, and we also found one individual who had a compound heterozygous variant that could not be detected and phased using short-read exome sequencing. So one particular gene that was very important here was this gene FGF14, which contains an STR expansion with a GAA motif, which when expanded beyond 250 copies, causes a recently described ataxia known as SCAR27B. So there's no current diagnostic test available for this in Australia. And so in our cohort of 34 undiagnosed patients, we actually identified 7 patients who carried a GAA expansion in FGF14 that is sufficient for a diagnosis of SCAR27B. And this was in fact the most common genetic diagnosis made across our cohort. It's important to note here that we also picked up 6 other patients who have expanded repeats in this gene that I'm showing here without the asterisks. But these repeat expansions either fall just short of that 250 base pair cutoff, or perhaps those repeats are not pure GAA, but they have other sequence motifs mixed in there, and those are currently not considered to cause disease. So once again, it's the ability of long-read sequencing to sort of fully resolve what's going on with this repeat. It gives us both the size, but also the sequence right across that repeat, even when there's mixed multiple motifs involved. So this is a really important advantage of this technology. What's also important to point out here is that there is now a drug for aminopyridine, which is also now recommended for patients with confirmed SCAR27B. So as a result of our analysis, all 7 of these patients where we found a GAA expansion of a certain size are now being treated with this drug. So it has real immediate clinical applications and advantages for patients and their families. To dig into another example here, we found a single patient who had 2 heterozygous variants in this gene SPG7. Now both of these variants are detectable using short-read exome sequencing, and this patient had short-read exome sequencing. However, these are both recessive variants, and therefore in order for them to be considered causative of this patient's disease, we need to show that they're on opposite haplotypes. So essentially we need to show that the patient has inherited 1 variant from their mother and 1 variant from their father. Now usually this would mean we would have to sequence the parents to confirm that. However, for late onset conditions like this, the availability of both parents for sequencing is often a barrier to resolving cases like this. But with our long-read sequencing, because we get long reads that sort of fully span this gene, we can phase this gene, and we can show that the 2 variants occur on opposite haplotypes. They're never on the same read. They're either on the blue reads or the pink reads, which means that we can show this patient has inherited 1 from their mother and 1 from their father without needing parental sequencing data. So this is, really streamlines this process for us, and is a really, low-hanging fruit in terms of applications for long-read sequencing, is just being able to resolve cases like this, which we otherwise just couldn't do with short reads. So overall, we achieved new diagnoses in a diversity of different ways. And I guess the take-home message here is really that this approach works well. This ability to capture all of the potential genetic causes of spastic ataxia in a single assay, which is a unique capability, I think, of long-read sequencing, clearly has capacity to streamline and improve patient diagnosis. So the technology works really nicely for spastic ataxia, but before I finish up, I'd like to quickly point out that this same strategy can be applied to any genetic disease or group of diseases, and there are many difficult genes and disorders out there where we think long reads are going to be diagnostically useful. So we're currently working on lots of different disorders, trying to develop sort of streamlined tests in the way that we've approached ataxia. So we're currently working on thalassemia, which is a relatively common genetic blood disorder caused by deleterious variants in the globin gene family. Now, because this includes numerous closely related genes and pseudogenes, and because structural variation in this locus is often involved, thalassemia is quite tricky to comprehensively characterize using short-read sequencing. And you can see here we get many gaps in the coverage on the short-read sequencing track, where we can't uniquely align reads to the locus, and so we get gaps in our coverage, and that's because of that homology between the different globin genes. But by contrast, we get nice uniform sequencing coverage when we use our long-read sequencing to target these regions. And it's basically the same story for this second example, which is the gene PKD1, which causes polycystic kidney disease. This is another relatively common inherited disease. It's a kidney disease, and this genetic diagnosis of this is unfortunately hampered by the fact that there are 6 pseudogenes that have almost identical sequence to PKD1. They're 99% similar, and so again, when we try to align short reads to this gene, we get a lot of gaps in the coverage where we can't uniquely align the reads. But by contrast, we get very nice even coverage across this locus using our long-read sequencing. I was going to show one more example at the end, but it's essentially the same story. Another very nasty kidney gene that we work on called MUC1 that you essentially can't sequence with short reads. But using our long-read sequencing technology, we're able to span through this very repetitive, difficult gene and ultimately build new diagnostic tools for this disease. But yeah, I'm going to just finish up by thanking everyone involved, my amazing team back in Sydney, the Genomic Technologies Lab, who are a wonderful group to work with, great diversity of skills and interests, and all of our collaborators. I mean, there's too many to show, but I've listed some of the important ones here, and yeah, very, very happy to take questions in question time. Thanks everyone for listening.